I'm gonna start my talk today. So my name is Chamseddine Kifagi. I'm senior scientist. I'm a specialist on chromatin biology in biomedical research, and this is my talk subject today. So I'm gonna start in this talk by some definitions on epigenetics and chromatin. After I will talk about the NGS-based analysis method, and I will focus after on Chipseek and ATAC-seq method. Those two famous or the main methods now on NGS chromatin-based analysis. After that, I'm gonna talk about three examples about chromatin-related project. And I will try to explain how the next generation sequencing and the high throughput analysis and technology advances has enabled researchers to better understand the impact of chromatin and on gene expression and cellular behavior and functions in normal conditions, in healthy conditions, and also in disease models. So I'm gonna start to define epigenetics and chromatin. So what is epigenetics? Maybe many of us know about of that. That epigenetics, it is mainly the heritable changes in gene expression without any changes in DNA sequence. Those changes are regular, natural, but can also be influenced by several factors like age, environmental, lifestyle, and disease state. So the epigenetic mechanism perturbation are very important and can lead and result in disease. The main mechanism of epigenetics are histone modification and transcription factors, DNA methylation, and non-coding RNA pathway. But in my talk, I will focus on histone modification, such as histone methylation, acetylation, and transcription factors. So briefly, I gonna make a brief introduction about what is chromatin. So the chromatin structure is based on DNA sequence of approximately 150 base pairs per base of DNA wrapped around 8 protein called histones. So this unit called nucleosome. And this unit gonna be repeated on the whole genome. That means on the whole genome we're gonna have mononucleosome, dinucleosome, trinucleosome, and etc. etc. So and what is more important that is this structure is highly dynamic. That means on the whole chromatin and on the whole genome we can find regions that there is open chromatin if for example this region expected to have gene expression and gene function, gene activity, and we have a compacted genome if there is no reason that we open the chromatin for the transcription, for example. And in that case, people talk about open chromatin or active chromatin and silent chromatin. So now I will talk about what can in the last 10 or 15 years ago with the new technology advances of a high throughput sequencing, the next generation sequencing, how the next generation sequencing allow researchers to go more in depth in analyzing chromatin. So here in this figure we can see different NGS methods of chromatin analysis, and all of them. So just the Chipseek target the transcription factors and histone for example methylation, but all the others can all target transcription factors and histones as well. So basically, all those methods start with protein or markers fixation on the chromatin. After that, we have to degrade the chromatin, even by mechanical degradation or fragmentation, like sonication, for example, or enzyme fragmentation. After that, we have steps of enrichment. If we use specific antibodies, we're gonna see that after. After enrichment, we have amplification of DNA sequencing and mapping of the chromatin. So in the rest of my talk, I will focus only on the main two methods methods called Chipseek and ATAC-seq. So Chipseek, it is for chromatin immunoprecipitation. So this method basically composed by 4 steps. The first one, it is to cross-link the chromatin. That means to fix the proteins or histone markers on the chromatin, on the DNA. And after that, we have the second step is to the chromatin fragmentation by sonication. We have after that the enrichment. That means immunoprecipitation by specific antibody and magnetic beads. So, sorry. For example, if we need to with Chipseek to focus on specific transcription factor or specific histone modification, we have to use specific antibody for that to just target this factor. And the last step, it is to reverse the cross-linking and to purify the DNA and go after with the DNA library prep and after sequencing. Yeah. After Chipseek, I'm gonna talking about I think the most new Chromatin NGS analysis method. So this method was discovered or was published in 2013 and called the ATAC-seq for assay for transposase accessible chromatin. So the difference between them, first, we don't need to fix protein by formaldehyde, and we don't need to fragmentate the chromatin. So this method is based on transposase, this enzyme called transposase enzyme. We use it to so this enzyme bind on the open chromatin and cut the chromatin and insert the adapters in the enzyme, and by this way we can just limit the nucleosomes and also the transcription factors binding sequences. After that, you can see on the figure on B. So after cutting between nucleosomes and transcription factors, we're gonna purify this DNA and go to the library prep and the sequencing. So this new method, it is highly cited now, and it is highly important mainly because two things. The first thing, that with this method we don't need a lot of cells to go to NGS. That means with Chipseek and other methods we need millions of cells at least. But with this method we need only a few thousand cells. And the other thing also, with the whole process and the whole experiment will be half of day compared to, for example, Chipseek will be 3 or 4 days. So briefly, I will this figure it is about the computational analysis pipeline, and on the top you can see that on the top we start with the FASTQ files from the sequencing, and after all the step of filtering, and alignment to the reference genome. And the last step, it is in the bottom. You can see the peak calling. That means this is this specific method and computational method to design what can be the peak of our factors or of our nucleo chromatin modulars, for example, or transcription factors. After that, after designing the peak location, you can see on the figure here briefly the steps from transcription factors. We have the million of reads, and we have the peak calling. And after that, many options and many we call that downstream function analysis are possible. For example, we can compare between two conditions, between two cells subset. We can see, for example, in 2 or 3 conditions or more, the intersection between the peaks. That means between the factor location on the chromatin. We can, after that, design or annotate the genes to those peaks, which are expected to be related. And we can also do transcription factor motif search and many heat maps and yeah. So now I gonna talk about 3 examples of chromatin-related NGS analysis. That mean on those 3 examples, I gonna explain as much as I can how the NGS and the high throughput analysis and sequencing allow to more understand and to more explain the role of the chromatin biology on some disease models. So the first example concerns the role of chemoprotein on the control of the chemotherapy of EML for ectodermal leukemia cells. So briefly, I will introduce what is chemo. So chemoproteins are ubiquitin-like proteins. They are known to bound on different proteins and also on different proteins on the chromatin. And the simulation and dissimulation pathway is completely controlled by different enzymes. And you can see here sumo enzyme 1 and 2 and 3. So those enzymes are expected to bind the sumo on the target protein. So the sumo pathway or the simulation or dissimulation pathway are known to be involved in many cellular activities such as cell cycle, DNA repair, transcription, and chromatin remodeling. And on the right side, this figure, it's about our project workflow. So we have here our happy cells, and in this project we treat those leukemic cells with specific drugs known to be so those drugs known to be used for the chemotherapy for patients, leukemic patients. So on those treated cells, we did 2 experiments in 2 sites. The one side we did the Chipseek experiment, and the other side we did the transcriptomic analysis. In the end, we overlap the results and the output of those 2 experiments, and the aim it is to clarify what kind of networks, what kind of genes, biological networks are regulated by this sumo pathway. So briefly, what was the result? The first result we can see on the left, that one of the most protein target on the chromatin of sumo was CTCF. And after that, you see the Venn diagram of our peak location between SUMO and CTCF, and also the intersection with transcriptomic results from many thousand peaks and for example, many hundred genes after transcriptomic. By this method, we can target on only 50 genes or 50 markers. After we can see that we did some experimental validation by qPCR and by cheap qPCR also, and we can see on the bottom also, this is the track view of one of our let me say best target, which is the NF-kappa B2. And on this target, we can see on purple a peak of SMC3, on red a peak of CTCF, and on blue a peak of SUMO. We can see, so this is, in this project, we start from the whole chromatin, from the whole genome SUMO location. We target on these peaks, SUMO peaks, CTCF peaks also, and we also target, so we also target some genes like NF-kappa B. On the right side, this is the gene ontology and the pathway analysis of our target gene list, and on this analysis of gene process, we can see that our SUMO target genes or our SUMO CTCF target genes are mostly inflammation-related process. So I will finish for this part of the project with to highlight the basic results of this project. So on this project, it was successful to see that the SUMO CTCF binding on specific chromatin regions affect specific gene expression, as I showed you, and that those genes are related to apoptosis and inflammatory process during the EML chemosensitivity. More interestingly, this what is more important on this project, it is when we inhibit or when we block this SUMO access, we can lead to EML chemoresistant. So moving to the example number 2, it concerns the transcriptional program of dendritic cells on the gut. So basically, I will briefly introduce the dendritic cells. So the dendritic cells, it's important and pivotal compound of the innate immune system in the body and in the gut. And it has an important role to move from the gut to the lymph nodes and to uptake the antibody antigens from the gut to the lymph nodes to present this antigen to B cells and T cells. So our project was mainly to see if there is a difference on the chromatin profile between dendritic cell subsets and location as well, and if there is a specific transcription factor program on those subsets. So briefly, this is the experimental workflow. So we can see here that we sacrifice number of mice, around 20 mice, and after we with fax machine and sorter machine, we can isolate our subsets of cells. We go after with the ATAC-seq experiment and library prep. On the bottom you can see the library prep, DNA library prep before going to the sequencing. And on this project, we try to follow the chromatin profile of dendritic cells subset CDC1 in different locations in small intestine, in lymph nodes, and in large intestine. So on this figure, I try to summarize the main results of this project. So the main results was you can see on left the Venn diagram of peaks, and you can see on the different location of dendritic cells in small intestine, MLN, and two parts of the large intestine, that almost more than half location are similar, and this is normal because all of them comes from the same cell subset. But in the same time, all data set have their specific chromatin location or chromatin peaks. That means that they have some specificity, and what we can see that this specificity, it is site-specific specificity of chromatin profile. And on the right, it is the heat map presentation of the transcription factor analysis of those peaks in the different in the 4 location of dendritic cells in promoter region and in distal regions as well. The most important thing that, and it is known also, but the most important thing, it is for transcription factors. They are some of them are more abundant on promoters, and some of them are more present on distal, which is normal. But what is more important here, and you can see on the red square, you can see that for NF-kappa B, one of known transcription factor, is more present on distal regions, but more present in lymph node than in small intestine. So here also result to see why we want to understand why this presence of these specific transcription factors are more present in lymph node. So we see. So here we analyze the target, the gene target of this transcription factor in MLN, and with gene ontology analysis and pathway analysis, we find in the top of biological process the cell migration, which is known to be one of the main function of dendritic cells in the gut. It is to migrate from the gut to the lymph node and to present the antigens to B cells and T cells. And in the right side, we can see with MLN, we can see the RNA-seq data also of only those cell migration targeted genes, and we're gonna compare with small intestine and MLN, and as we can see here on the right, that we have a couple of those targeted genes are upregulated in MLN compared to small intestine. And also on the right side, we can see 2 examples of those targeted genes, and you can see that on the chromatin, on the DNA, that we have on blue peaks, that we have a peak on MLN, and this peak is disappeared on small intestine. So the main results of this project, it is 2 questions, 2 biological questions, is that the migration of dendritic cells from the gut to MLN can be derived by NF-kappa B binding or chromatin, and is that related, for example, the cell migration of those dendritic cells? And those results are now going to be validated on the lab. I will finish with this third example. It's focused on specific translocation on leukemia called MLL rearranged leukemia. So and this team, Armstrong team in Harvard in States, discovered maybe around 10 years ago, discovered that this specific translocation lead to the highly presence of this protein .1L on a specific region on the genome, and this high binding on itself also promote the high methylation of the histones by specific histone methylation marker called H3K79 demethylation. So this nice discovery was highly and mostly cited in this field in leukemia. And after that, this team go more in depth in their discovery and go and develop specific drugs to inhibit this protein binding on the chromatin. So those specific drugs target the binding of .1L protein on this specific region on the chromatin. And after that, now they are going and after that, so this drug is now in I think in phase 3 of clinical trials. So this is a nice example to show you how to go from very fundamental finding of chromatin involvement in disease model, and to go to develop what we can say now the epi drugs in leukemia and in cancer disease. I will just finish with what is now what is next after the next generation sequencing, and what is the next challenges. So the next challenges now, it is the single cell chromatin profile. And 3 years ago, this thing it is or 5 years ago, this thing it is possible. And one of the high one of the big company of the single cell in the world, 10x, they are going that now. They have on their panel the kits and the knowledge of single cell RNA-seq. This is a nice example of a very new research paper, and they describe how we can on the same few cells, number of cells, we can do single cell RNA-seq, single cell attack-seq analysis, integrate them, and go in depth and see what kind of molecular mechanism can be seen in specific disease models. I will finish with this slide to say that I'm a member of a nice and huge network of young researchers in biology in Tunisia, and this network expected to share science, to lead science, and to promote the science in our countries. And we gonna organize international symposium every 2 years. It was expected to be done on last March, but the event was postponed to the next year. So all of you are invited to follow us and to massively participate in our nice event. And thank you. I think I'm in the time, maybe a few seconds before. So thank you very much.